Hello, everyone, and welcome to phyloseminar.org. This is the first talk in a series of 3 talks on the intersection of phylogenetics and public health. Please use the YouTube live comment box to ask questions. Today's speaker is Henry Kunerth. Henry completed his PhD in ecology and evolutionary biology at Cornell University in 2022. There he studied the evolutionary genomics, population genetics, and speciation of an invasive moth, the European corn borer. After that, he made a jump into the world of public health, joining the Minnesota Department of Health Infectious Disease Laboratory as a research scientist and bioinformatician. Broadly, he has interest in applying insights from his PhD studies to better understand infectious disease outbreaks and how the genomes of the pathogens that drive these outbreaks might be shaped by natural selection. He also has strong interest in teaching, which he currently applies by disseminating methods and best practices learned in public health to help educate a new generation of laboratorians, bioinformaticians, and genomic epidemiologists across the globe. Welcome, Henry, and thank you for participating.
Thanks, Eric. Yeah, I'm happy to give this talk. It's as I was looking back at some of the previous speakers, it was fun to see sort of the intersection of my past in kind of a pure research scientist capacity during my PhD to what I'm doing now, which is much more applied and a little bit different than my past, but it's kind of fun to be able to give this talk and unite the two interests. But before I begin, I just wanted to, I'll see if my slides advance. Just wanted to say a quick word. So I put a map here. So we're here in the Minnesota Department of Health. I'm actually down in this building down here. And this is the Minnesota State Capitol, so it's just a kind of a stone's throw away. And if you follow the news at all, this is basically what it looks like when I walk into work from my parking lot down here each day. If you follow the news from the past weekend, Minnesota State legislator was assassinated in her home over the weekend. So I guess I just wanted to kind of open with that and take a minute to pause and reflect on that, because it's been a tough time in general being in public health the last few months, but especially so when essentially a colleague of mine, I didn't know her, but was killed. And so, yeah, again, kind of just keep us in your thoughts, and yeah, bear with me if I'm a little spacey today. But with that, I'm going to move right in. So again, being able to introduce this public health circuit on the phyloseminar group, I just thought I'd give a little kind of intro to how Department of Health is organized generally. And I'm going to zoom in on where I'm sitting, which is in the Infectious Disease Laboratory at Minnesota, and then point out that we've got a number of different units within this department that are doing a variety of different things, and we have communication between them. But we're all, kind of in some way specialized. So we have everything from emergency preparedness and response, responding to, say, like a bioterrorist attack or being prepared to do so, to enteric diseases, microbiology, health informatics, the people kind of keeping all of our databases connected, virology, operations, scheduling, shipping, receiving, budgeting, that sort of thing. And then where I sit, which is in the sequencing and bioinformatics side. So as Eric mentioned, I'm a research scientist and bioinformatician here. So a big part of my day-to-day job is constructing and maintaining bioinformatic pipelines so that we can, anything that we sequence can kind of quickly be moved through from any of these other departments through the sequencing process into sort of actionable data that we can then pass on. And we pass on and have dialogue with a lot of different groups. So this is just a sampling of the different people we communicate at the health department, but it's certainly not an exhaustive list. But we have partners at the CDC. We talk to a lot of clinicians and people at hospitals. They send us samples. We send them back what we discover. National and global organizations like the American Association of Public Health Laboratories. Our academic partners, we have a number of different grants that are collaborations with partners at the University of Minnesota, the Mayo Clinic, beyond there. This is sort of within the health department, but we speak a lot with epidemiologists and try to discern or disseminate, I should say, our genetic data into data that they can then use and take to the field for interventions. And we also communicate with tribal leaders groups. Again, not an exhaustive list. I'll just point out again. So I'm taking this perspective from the sequencing and bioinformatics side, but all of these people are involved with this broader effort to improve and tackle public health challenges at the state and national level. So zooming in on our unit, here's our group. We're Sean here was our former supervisor. He's left now, but everybody else is still doing this day-to-day work. It's a team of mostly laboratorians. There's 2 bioinformaticians, myself and Jake Garfin, and then Dan Evans, who is kind of central to this talk. I'm going to circle some of the people involved in the human respiratory syncytial virus core group. Sarah really got the project going. Matt and Laura are doing a lot of the lab work and sequencing for this project. That's me in the back there, and next to me is Dan Evans, who's a genomic epidemiologist, who is a major contributor to this talk. A lot of the slides I'm going to show and talk about are his. I know he would have loved to have been on this talk with me, but he's on vacation this week. So we wish him well, and really a huge thank you to him for his contribution to this project. Okay, so with that kind of introduction in mind, I'm going to get right into the specifics. So the project that I want to talk about today is one that we've been working on for a couple of years now on human respiratory syncytial virus, or hRSV. I might shorten it to just RSV, but we're talking about the type that infects humans. This is a virus that causes disease in essentially all groups, but the risk of severe disease is much higher in elderly, young children, and immunocompromised persons. In Minnesota each year, we see about 3,000 hRSV inpatient hospitalizations each respiratory season. The virus, I'm not a virologist by trade, so any specific questions about the virus, I'd probably have to pass on to a true expert. But it takes its name from the formation of the syncytium, this sort of fusion of cells that allows for reproduction, more efficient reproduction within this sort of larger host cell. As you can imagine, that's not something that typically the human body will want to be doing, so that's part of the contribution to disease. But you'll have this infection, then a fusion with other cells, and then reproduction, and so on. This is an RNA genome. It's about 15,000 nucleotides long. It's single-stranded, and it, in many ways, is fairly similar to the SARS-CoV-2 genome. It's about half the length, but in otherwise is quite similar in that it's single-stranded. It's an RNA genome, and it's quite small. And so a lot of this work was built on all of the extensive work that different groups, including ours, have done since 2020 in developing sequencing infrastructure for SARS-CoV-2. And so we kind of had this thought going back a couple of years that we could take a lot of that infrastructure, the sequencing techniques, the machines, the bioinformatics, and apply it to a different widespread respiratory virus. And provide some of the same sort of actionable insights that we, even if you're not, intimately involved in public health, I'm sure you've seen over the past few years, a lot more reporting than you would have otherwise ever seen in your life about, the nitty-gritty of the SARS-CoV-2 genome, different lineages emerging. You'll see that on front page news in the past few years. That really wasn't the case before. So we thought we could do similar with RSV and hopefully, better tackle this virus in the state. Last thing on the virus itself, there's 2 major viral subgroups, hRSV A and B. So I'm going to refer to these as RSV A and RSV B throughout the talk. They're 70% of their genome is almost identical, but they do have some key differences. And these genes in particular, the G and the F protein genes, these are genes that are involved with cell recognition and fusion. So they're the biggest targets of selection and what vary most between the A and the B subgroups. So in terms of surveillance, sort of before we get to the genomic epi side of things, and sort of what existed before, just a few kind of caveats and carve-outs here. Generally, this isn't a reportable virus, so we don't get all, there's some viruses that are required by law to be reported to the Department of Health if they're encountered in a clinical setting. That's not the case with RSV. However, we do have a program called RSV Net, in which hospitalized cases and associated deaths are reportable through this program. So we get word on basically the most severe cases. And then we also, can call and ask for voluntary submissions of the less severe cases. Generally speaking, especially before we were doing any genomics, epidemiologists rarely received any even A and B subgroup data, much less anything less granular than that. It was essentially, is a patient positive or not, via a PCR or an antigen test. This really limited outbreak response and meant that there was no genomic analysis involved with it. So if there was an outbreak, you might see an uptick in cases that experienced epidemiologists can do a lot with, but there's nothing more granular that's really hard to trace, origins of outbreaks, things like that, without or prior to the advent of genomics. So this is where we wanted to kind of begin to fill that gap. And so one of our major questions here was, can we collaborate across this sort of lab that's us in the Infectious Disease Laboratory, the lab epi, the epidemiologists who sit in the building next to us, across this lab epi interface. It's kind of a buzzword in public health, in order to strengthen RSV surveillance by using whole genome sequencing. So what does whole genome sequencing allow us to do? Well, it allows us to be more precise in our surveillance, be more specific. We can detect clusters and outbreaks and then ask if they are related, to one another. We can identify new variants and lineages and better inform the public by using, terminology that is more understandable and digestible to non-scientists. And then we can try to take that a step further and identify specific mutations or any subgroups of the virus that might be associated with phenotypes such as, higher disease risk or vaccine escape or monoclonal antibody resistance, things like that. So as I mentioned before, we adapted our SARS-CoV-2 laboratory protocols that we had, developed since 2020 in order to sequence and analyze these hRSV genomes. Across the past 2 years, we've received hRSV positive specimens from, I think it's upward now of 29 healthcare facilities in Minnesota. It's at least 29, I can guarantee that. And we took a tiled amplicon sequencing approach. So if you're not familiar with this, this basically involves amplifying short segments of the genome, as you can see represented by these arrows, that have overlaps with one another and and span the entire genome. So we had 50 total amplicons across the 15,000 base pair genome with overlaps across each. And as such, assuming all of these amplify, we get a complete picture of the genome after we put it together using a pipeline. So in terms of that pipeline, if you're interested in the details, I'd be happy to communicate after the seminar. You can shoot me an email. But generally, we did the assembly, QC, and typing using a NextClade, a Nextflow viral recon pipeline, some slight modifications to it, and NextClade for assigning type. So there's different ways you can type things. You can assign things at a very sort of high level to a subgroup A and B. you can zoom in on specific glycoproteins. So this is the G protein here. This was a classic target for typing. Or lineages, which uses basically mutational data from the entire genome. So I'll mention that more in a little bit. And then I mentioned my colleague Dan Evans. A lot of what he does is develop phylogenetic trees using the next-strain auger pipeline, turn that into, and basically turns that into data that can be passed on and understood by genomic epidemiologists such as himself. In terms of the primers themselves, again, I won't dwell on this, but I thought it might be of interest. There's a group led by Danny Maloney in Scotland who developed basically these Arctic style, very similar to what a lot of labs are doing in terms of SARS-CoV-2, Arctic style amplicon sequencing for both RSV-A and RSV-B. And one development here that was pretty nice on the lab side is we can multiplex RSV-A and B together. A lot of the samples that we receive were agnostic to whether it is A or B. So it's nice that we can just take a single sample and load it with a multiplex reaction of both A and B primers and then sort out the assembly in post. It does cause occasional complications, but I'll mention in just a little bit.
So, Henry, I know this isn't necessarily your wheelhouse, but I'm just curious, what does Arctic style mean? I know that they do tiled sequencing, but that's about all I know.
Yeah, so the Arctic protocol, as far as I know, and I came into this, so as Eric mentioned, I finished my PhD in 2022. So I kind of came in right after the real challenging part of being in public health during the COVID response. But I think that the Arctic basically primer scheme was developed for widespread sequencing of SARS-CoV-2 using that kind of tiled amplicon approach. I'm not sure where it got its name.
No. I just meant okay, that's fine. I mean, it sounds like it's a tiled amplicon sequencing setup, so that's great. We can move on.
Yeah, we can chat later. I might have misunderstood your question.
Great, thanks.
Yeah. And then, so in terms of assigning lineages, we define them based on a recent publication by Stephanie Goya and this basically this RSV consortium group, which is, if you followed at all the way that SARS-CoV-2 lineages have been assigned using this approach called pangolin, it's a similar style approach of assigning names. Well, the community tends to be the one that kind of assigns the fun names, but assigning basically alphanumeric names to RSV genomes at a level below this subgroup. So in terms of our work, so from July 2023, which are really kind of when we began this program, through May of 2025, we generated over 3,000 complete genomes in roughly a 50-50 distribution of RSV A and B. That's purely coincidental. We definitely didn't filter on that at all. In general, kind of our high-level findings, we did see some differences in terms of average genetic distance between the subgroups A and B. They're definitely distinct. I'll zoom into some of that data a little bit later. At the moment, we're still lacking data in order to associate lineages with the severity of disease. Part of that is what I mentioned earlier, where we only get data, or sort of the only required submissions are hospitalizations. So some of our data set is kind of skewed in that direction. If we had kind of a widespread surveillance network, like you might recall from the early days of the COVID pandemic, we might be able to have kind of a better baseline for lineages that may not be associated with severe disease. But right now our data skewed towards severe disease, so it's kind of hard to associate lineages with that without kind of a proper control. And then this is one thing that I wanted to mention if you're thinking about doing any of this work or just thinking about understanding it. In our data set, it's a small number, but something like 1 in 200 of our genomes that we sequence, we see evidence of a potential co-infection of A and B. And really that only turns up at the kind of in post as we look at the assemblies, and we have to be pretty careful about catching it because, as I mentioned, we generally don't have a type assigned for a sample, and then we multiplex a single sample with A and B primers. Now, if that individual happened to have both RSV-A and RSV-B, they were infected by both of them, and therefore, we, had viral particles from both A and B, we would amplify both A and B sequences, and we would assemble, without knowing, that we would assemble essentially kind of a mismatch of hRSV A and B. That leads to some QC issues, and it's something that I've observed in global databases. So it's something we want to be really cautious of, but it's also interesting too. We're not seeing, sort of once you're infected with one of these, that virus the individual seems still susceptible to the other type. So it's something we want to follow up on. We're kind of curating this data set, and we haven't done too much with it yet. So here's our picture across multiple respiratory seasons. So the 22-23 is a really limited window, kind of, as I mentioned, we started in July 2023. So that's really the tail end of that respiratory season, which we generally define as beginning in the fall of the year, September-ish through May. And then we have kind of a, RSV, other than rare summer outbreaks, tends to be fairly quiet in the summer, like a typical respiratory pathogen. But you can see in these time-scaled phylogenies, the three different seasons that we're working with. In green are the 2023, 2024 samples, and then our most recent from 2024, 2025. And this is hRSV-A on the left and B on the right. I'm going to zoom in on these in a little bit, but I just wanted to point out some general features, which is, we're observing the emergence of seasonal clades, like this one here. This really wasn't present in the prior year, and then it really exploded, and roughly half of our RSV-B data belong to this clade in the 2024-25 season. And then we're also seeing a loss of clade during what we're inferring to be, a summer bottleneck. So these were widespread clades in 23-24, and then they left no descendants in the 2024-25 respiratory season. So that's pretty interesting to us as well. Another thing that we can start to do is link epidemiological data to RSV genomes. So this is from a paper that Dan and I contributed to last year. So it's a little bit more limited in scope than the broader data set. But I just wanted to point out kind of some of the ways that we link this data. So, we can look at changes across the season by, looking at when these samples were collected. This bar here is basically a breakdown of the different lineages. As I mentioned, we have this RSV net program, which basically flags data from the more severe cases, or what we see as a proxy for the more severe cases, because they resulted in hospitalization or death. If you were really sick with RSV and never went to a hospital, you'd be missing in this data, of course, so it's limited. But we can basically say these cases seem to be the more severe ones, and anything in the gaps might be, someone who went in because they were sick, got swabbed, we got the swab sent to us and sequenced, but they were maybe dismissed and went home to ride out the infection themselves. So it's kind of a proxy for a less severe case. So we can start to ask questions about, are any of these lineages, do they have stronger associations with what we see as more severe disease or less severe disease? Again, it's kind of in the early stages. We want to build on another respiratory season in order to sort of make any of those predictions a little bit more concrete. And then we have some other data. It's really limited at the moment, but any patients that were administered nirsevimab, an antiviral, and whether they were in a more severe case or a less or potentially unknown severity case. And then we can also start to map on specific mutations like this F protein K68N, which is a mutation associated with monoclonal antibody resistance, and see if this is emerging across season in any of the other lineages. So these are the types of questions that we're asking. Just a quick word on this nirsevimab breakthrough case. This is nirsevimab is a monoclonal antibody administered for short-term immune boost against RSV infection. And there are some known RSV genome mutations that confer resistance. They basically, here I'm going to zoom in. So we did have 2 RSV-A hospitalizations occur after this administration. And so we were particularly interested in these genomes. Again, it's a super limited sample set, so we're kind of keeping an eye on this sort of thing moving forward, rather than drawing any specific conclusions. But if we were to see an uptick in hospitalizations after nirsevimab, we could investigate those genomes for any of the known resistance mutations. And we did see, again, this isn't a case of a high degree of hospitalizations after or in this group, but we did see a clade of RSV-B genomes that shared this mutation that I mentioned earlier. So as I continue this kind of tour through how we're using this data, we're also taking a look at how Minnesota genomes compare globally. So RSV is a globally distributed virus. It's widespread, and we tend not to see super localized patterns. But with the amount of sequencing that we were doing, we had this unique opportunity to really kind of test some of those hypotheses or predictions. So what we have here are 2 phylogenies, hRSV-A on the left and B on the right. This is again from our 2023-2024 data. I'll show some more updated data later. But we can start to see where the lineages that we collected in Minnesota fall relative to the global picture. So anything marked in the black color is from Minnesota. Anything in the kind of gray is a North American sample that's not in Minnesota, and anything in the lightest gray is a global data set that we pulled from GISAID. And you can see there are some clades that seem to be highly specific to Minnesota. And I guess I should caveat that and say they're not necessarily specific to Minnesota, but we're doing so much sequencing that we're detecting them, right? So I have no doubt that these, lineages, if they are geographically associated, then they're geographically associated, say, with the Midwest or the Central United States. But since we're doing so much sequencing, we're identifying them, and they appear, Minnesota specific. I'll just be careful about kind of using that localized term. But I will say we're doing just an enormous amount of sequencing. So since August 1st of 2023, we have, I mentioned this figure before, sequenced roughly 3,063 full-length genomes that passed all of our QC. And that's roughly 19.5% of all the sequences, the complete sequences in GISAID as of June 6, 2025, in that same period. So it really is an enormous amount of the kind of global work is coming out of our lab. And I don't just say that to toot our own horn here, but more to just say that it's to thank a bunch of people who have contributed to this project, but also to say that it's kind of this unique opportunity to test some predictions about what that means. So these are genomes specific to Minnesota from 2023 on. It's a little over 1,500. The outer or this inner circle here represents the sampling year. So lighter is earlier. So you can see, we've done more and more of this over time. There's more of the darker color. And you can see the emergence of some of these seasonal clades like this one here that, we really didn't see any evidence of prior to this big outbreak this year. And then we're starting to fill in some phenotypic data on the outer edges here. These are genomes associated with known monoclonal antibody resistance mutations. And you can see that, they do cluster into certain, smaller clades that contain them. That's kind of no surprise, but is interesting for us to follow up on. The same thing for RSVB. About the same number. And again, the emergence of these seasonal clades like this BDE 1.5, which is, almost, well, it is entirely this year that it was identified and given this name. So we're kind of tracking this lineage, and it's really only showing up in our data as well. This is kind of the rough picture of when we are receiving these specimens from each of the past 2 years. As you can see, we've really increased the amount of samples that we're receiving. We basically have been trying to ask as many submitters as possible to send us all of your RSV data. We really want to sequence it. This is what it breaks down to roughly by the subtype. Like I said, it's a almost kind of eerily 50-50 split between A and B. Although we did observe this year for the first time a real spike of B earlier in the year, followed by a spike of A later in the year. That is something we want to keep track of. Last year, they really tracked each other with just a couple of small blips. Then if we break this down, as I mentioned, by the lineages that were defined by the Goya et al. Paper and group, we're seeing the emergence of new lineages across each season. So, if you can just kind of eyeball these plots, you can see that, we're getting more colors in this second year in addition to just more data generally. And the same in this next season. And it was really driven by the emergence of two new clades, B.D.E 1.5 and B.D.E 1.6. Kind of a mouthful, but they emerged in our data in this most recent respiratory season, and we're keeping an eye on them. I'll highlight, one of these clades here, or actually two of these. These are the ones I just mentioned, B.D.E 1.5 and 1.6. These were really uncovered by our sequencing data. They're not really if you look at the map on the right, they don't occur outside of the United States, and it's really, just our work that's uncovering them. We're interested to see if that, trend continues, if we're able to continue to do this amount of sequencing next year. It I think stands out in terms of the global picture, the amount that we're doing and what we're seeing. We also are observing, that not every year we're seeing the same, genetic patterns. So this is just a plot of pairwise SNP distance between each sample in our data set. And, they're not the same year on year. I won't ask you to kind of squint and eyeball this plot too much because it's essentially being driven by the seasonal emergence and loss of new clades, that have an approximate genetic distance to the other clades in the data set. That's essentially why these kind of peaks are shifting year on year. Same thing as well, if we kind of look at how clustered our data set is, basically the distance from the nearest neighbor, roughly, 80% of all of our genomes end up, being quite closely related to, a neighbor within, 5 SNPs or so. And only a few have very tight associations with the nearest neighbor, and they tend to follow the same trends year over year. I mentioned before, the emergence and loss of some of these clades. So we'll go back to this figure here and say, we're observing the seasonal loss of some clades during that summer bottleneck. At least that's our sort of working hypothesis for why some of these clades would be winking out over time. RSV numbers tend to drop over the summer quite a bit. Whether these clades had any, phenotype that made them less prone to survival over the summer, or whether that was, purely sort of drift is a testable question, but one that, we're still working on. And the same thing here with the seasonal emergence of these new clades that we really weren't seeing prior to this year at all in any of our data, whether it was Minnesota or the global picture. These, obviously have near relatives. They didn't come out of absolutely nowhere, but we didn't see anything quite like them. So that's something we're also keeping an eye on. I'll just point out the same for RSVB, essentially the same patterns. A few clades get lost each year, and then a few major clades have emerged each year and really taken off. I'm kind of nearing the end of the talk, but I did want to mention an ongoing project. I don't have tons of data to report on this yet, but I wanted to bring this up and also ask if there's anyone on the call who has, ideas for how we could use this data. I'd love to hear them because, this is an ongoing project. So I'll just mention a few of the things that we're asking and working on right now, and a couple of tiny bits of data that, again, is still ongoing. But we're observing these seasonal changes in the clades and lineages. We have access to a lot of high quality data, not just ours, but all of the people who are contributing to, RSV genomes to the global databases like GISAID and NCBI. So, we can start to ask questions, for example, although I'd like this list to expand, can we find evidence of any site-specific positive selection in RSV genes? So in order to begin to answer that, I pulled complete, meaning, greater than 14,500 base pairs. The RSV genome, I'll remind you, is about 15,500 base pairs. From these past two respiratory seasons, so September 2023 through May 2024 and 24 through 25 from GISAID. I removed anything with more than 10% missing data, then produced gene by gene alignments and did a lot of manual inspection of the data. One of the things that tends to sneak through are these instances of, I think, are likely contamination of A and B, or, contamination or co-infection of samples where we're basically assembling a little chunk of RSVB into an RSVA genome. They really stand out, and, you can grab those snippets out and see that they, match the other subtype. It's easy to see once you see it, but hard to detect before that assembly process. And then I produced these 5 data sets, each for RSV A and B, for each of these genes. And basically, I broke these down by the same subdivisions we were talking about before, global, North American, and Minnesotan. So this is kind of our complete genome. And then we can pull out just the North American genomes. Then we can pull out just the Minnesotan genomes. And then we can also remove our Minnesota data from these data sets. And these all allow us to ask different questions that I'll run through right now. So, we have different questions kind of for each of these different data sets. One, we can ask questions regarding selection. How are the global picture in general shaped by selection? Or how does selection shape just North American genomes or just Minnesotan genomes, right? So these are kind of high-level selection questions. And there we go. We can also ask questions like, are there sites that are under selection in North America in this genome, but that are not showing up in the global data set? So basically, are there any sites that are really driving or, showing evidence of selection in this data set, but not in the rest of the world? Site, localized, phenotypes, basically, or the same thing with anything in just the Minnesota data. And we can also ask some kind of methodological questions, or more, I guess, sort of impact-based questions. Do we see sites that are affecting the global picture? So, if we were to uncover sites in this data set that do not show up in this data set, we would basically be able to say, well, our contribution allowed us to, find this site that might be under selection in this data set. Or the same thing here in the North American data set. Did the contribution of all of these samples, the 20% of the global samples, lead us to new insights, just to be able to, say what impact we're having. We can also take this and generate year-on-year comparisons. So, for example, with any of these data sets, how do any of these sites that are under selection change with each respiratory season? And is selection shaping lineage survival and loss? That question that I mentioned before. Just as a first pass at this, again, I'm in the early stages of doing this sort of year-on-year analysis. I did some of it for last year and then adding this year's data right now. We're basically taking approaches, widely available in the literature. We're a little bit limited in, how much we can do as a public health lab with, other responsibilities. So I'm kind of not trying to develop any new methods for this data. I'm taking the stuff out of the box. So for example, using approaches like FUBAR as unconstrained Bayesian approximation for inferring selection, looking at anything that pops up with as a first pass with the Bayes factor over the 15. And I'll just show a little vignette of some of this data. I am in the early stages of this. But if we look at the hRSV glycoprotein gene, this is a gene I mentioned before that has a function in viral adhesion. It's a very common selection target, has by far the highest nucleotide entropy across the genome. If we look at this global data set, we're uncovering 24 sites in this gene, across the 300 some individual codons in the gene that seem to show evidence consistent with natural selection in 23-24 across that season, and slightly fewer of them across the next respiratory season. But 10 of these notably were shared across the respiratory season. So if they are in fact under being shaped by natural selection, it's an ongoing effect across the season. And kind of equally interesting to me are anything that did not show evidence of selection in one season, but did in the other. Kind of a similar picture here if we zoom in on this North American data set, slightly fewer hits, but a similar pattern of 18 in the first season, 12 in the next, and then about the same fraction of them were shared. And again, we can start to do more comparisons, how many of these were shared across this data set and this data set, et cetera. And then a limited number in just the Minnesota data set. Again, these data sets get smaller with each reduction, so it does make sense that we'd be picking up fewer and fewer hits, but a couple of them that were shared across the respiratory season. And then again, if you go to that other sort of more methodological question, what impact did the contribution of Minnesota genomes have to our inference of selection? We do see that without these Minnesota genomes in the picture, we detect fewer sites in this global data set, dropping from 24 down to 21 and 14 down to 6. And then if we look at North America, going from 18 down to 15 and 12 down to 7. So the addition of these Minnesota genomes is, at least with this approach, it's, this one narrow approach, does seem to be affecting what we can uncover on a global scale. So again, just a tiny little snippet. I'm going to continue to work on this, and hopefully we'll be putting some of this data out in a preprint in a paper coming soon. So I'll kind of begin to wrap up and ask some, or say what I think this means in terms of public health. We know that this work in Minnesota is shifting this global landscape of the study of RSV. We're seeing that in the emergence of clades specific to Minnesota. We're pairing this with RSV-Net data and trying to categorize our data as severe or potentially more mild or moderate in order to look for phenotypes that might be contributing to the severity of disease. We're generating data for these genetic studies of RSV mutations during and between infections, looking into that further. And we're hoping that some of this work influences strategies for studying other viral pathogens. Ideally in the future, we'd kind of have a unified respiratory pathogen panel in order to sequence anything that if you have a cough of any type, you can get it sequenced and add that data to these sort of data sets. We're also seeing, early stages of evidence of positive selection in these genomes. Again, there's more to come. We're going to keep working on that. We know that we're adding to this global picture, and every year we're looking to expand our sampling to a broader geographic and age distribution. I didn't mention it specifically, but one thing that was limiting us in that first year was, a large bulk of our specimens that we were receiving were from the Minnesota Children's Hospital. As such, the age distribution of the patients that were infected with RSV skewed very young. But one kind of success of the second year was we were able to reach out to a lot of other submitters, local clinics and hospitals, for example, throughout the state. And the next year, our age distribution really smoothed out a lot more. It still skews young and old because those are the people who tend to be to really notice their RSV infection and go in and get tested. Yeah, we're going to continue our efforts to expand that sampling so that we can capture, as much of the RSV that's circulating in our state as possible. And then thinking about our future directions, every day we're trying to think strategically about how to use this data. As I mentioned, I'd love any feedback from anyone on this seminar for, different ways, now that I've kind of moved away from an academic setting for a couple of years. I'm not necessarily thinking in terms of some of the directions that we might be able to take on a pure science level. So I'd love to hear any fresh ideas. But in terms of the more applied, steps that we can take, can we, use this data to prospectively identify and intervene against RSV outbreaks, or can we get our surveillance down to, a granular enough level that we can maybe catch and stop outbreaks before they happen? I've mentioned this a couple of times, but can we identify any phenotypes that might be linked to severe infection and see if those are being shaped by selection as well? And then can we track any emergence of new mutations that might be linked to specific phenotypes like antibody resistance or vaccine escape? The RSV vaccine has really only been widespread or sort of available in a widespread setting for just 2 years now. So which coincides with our data. As we get more data about, vaccine uptake or vaccine status of any of the patients that we're seeing this in, we might be able to make some inferences there. And then, can we motivate any other public health labs in other states to not just sequence COVID or SARS-CoV-2, but, begin to sequence other pathogens? Well, plenty of states are sequencing other pathogens, but do kind of similar COVID or SARS-CoV-2 style sequencing programs for other viral pathogens such as RSV. And kind of add to this growing data set. And this is a bit of an outdated question, actually. I think this slide was originally put together when we just had the 1 year of data, because we definitely saw we can answer this question affirmatively that, yes, I don't know if it was us who motivated them, but other states have, done similar work to what we've been doing, especially in this past year. With RSV, we've seen that in the data. So yeah, with that, I'm gonna wrap up and take questions. I know I think I'm a little ahead of schedule, so maybe I'll have time to answer a few. But again, this is our group. And if you want to reach out to me, I'll leave my email up here for a little bit. If you have any questions or ideas, I'd love to chat. And also, Dan Evans, again, back left here, is the genomic epidemiologist who's done a ton of this work as well. I wish he could be on the call today, but he's definitely a great person to reach out to as well. So with that, I'll thank everybody who's listening and thank Eric for inviting us to give this talk. It's always fun to talk about this data.
Really exciting. Thank you very much, Henry. I have a couple questions. Well, I guess the first thing to say is it is exciting to see all these things that were developed for SARS-CoV-2 getting repurposed. We have Nextclade. We have Pango.
Yep.
We have Arctic style things, and this is, I think, evidence of the hope that, you know, when we had that big bad thing happen, that some good would come of it.
Yeah.
Can you say a little bit more about what the scope? I don't know RSV enough to know what is the scope of interventions. I mean, when it was sort of the height of COVID, there was sort of like a menu of possible interventions that people could recommend. I don't know what is the scope of possible things one could try to do to reduce the spread of a specific lineage.
Yeah. I mean, I think it obviously it depends on, I suppose, receptivity to those interventions, right? That's a big factor, like COVID. And at the peak of the COVID pandemic, the ongoing COVID pandemic, there were, for example, if we had an outbreak detected in, a nursing home, and it was confirmed by sequencing, there was a lot of work being done to trace the origin of that outbreak, to apply extra say masking or social distancing programs, I suppose, to limit that spread and stop it. Or we've seen outbreaks in workplaces and schools and such. And I think we would hope that we could see similar here if we were able to detect an RSV outbreak in a similar setting. We're probably more limited in terms of there just isn't an RSV surveillance system similar to, and really there isn't one for SARS-CoV-2 at this point anymore, where people are just doing routine testing, weekly testing or biweekly testing in schools or nursing homes anymore. So that definitely limits things. And instead, we're just seeing samples basically from people who go into hospitals or clinics because they're already sick, which makes it harder to intervene. Yeah, I don't know. I mean, that doesn't really answer your question that well, but yeah, I don't know. I'm also not an epidemiologist, and I'm sure if there's epidemiologists on the call, they're like, we do this thing all the time, and I'm just not saying it. But yeah, I wish Dan was on the call because he could answer that better.
Yeah. So it's interesting. So I guess one thing I'm just curious, like, who is doing the Pango? Like that requires curation. So is that something you all are doing?
Yeah. So there's this RSV consortium that has Stephanie Goya published that paper. I know. I'm not sure I'm going to get the pronunciation right, but Richard Nair, who I believe gave a phyloseminar, I think I saw in the past. I've communicated with him, and a lot of people who are, I assume, volunteers are curating that page. In terms of it does seem to be voluntary, and there's a GitHub page where you can submit lineages or potential proposed lineage designations with specific mutations. And I know there's that voluntary submission portal, but also just ongoing work by people in that group looking at the national and global databases for emerging clades. So I feel bad about this, actually. We probably should have volunteered that clade, some of those clades that we really saw emerging in Minnesota, to them. They were able to identify them and stick labels on them without us needing to ask. So credit to that group. But yeah, when we saw them emerging, we probably should have been quick to ask for them to get a new designation, but they did get one in the end.
So when you're, is there a chance of people sending samples to Minnesota? Or maybe that's even just hard to like move stuff around between states, but it just seems like you all are so good at doing this RSV sequencing that And it would be neat to, I mean, like the contribution you've made is awesome. I mean, that's a huge percentage of the sequences, but if you could have that percentage that was also dispersed across the US, that would be.
Yeah. I think possibly next year, assuming, we can in terms of our, legal and contract and, state jurisdiction and all of that sort of stuff. It would be great to get samples from states who maybe don't have the capacity that we have, but would allow us to expand our scope. Basically, the 2 years were, the first year was just, we'll take anything we can get. I think we had like 9 total submitters, and like I said, a bulk of those being Minnesota Children's Hospital. And then the next year, we gave, actually one of the things Dan's working on is kind of trying to plot a timeline of when we gave talks, really begging submitters to give us more samples, and then whether we saw an uptick after that, to kind of see if we can quantify all of our requests a little bit. But the next year, like I said, it was really focused on expanding past those 9 and really trying to, make children's hospital submissions, keep them level, but make them a smaller proportion of our total samples statewide. So going into the next respiratory season, assuming we're able to continue doing this work, with funding uncertainties that we're all dealing with in the academic and public health world, I think it would be great if we could expand outside of our state. I'm not, I guess I'm not willing to be confident about that just because of kind of the broader picture of what funding looks like. But yeah, it'd be great.
Do you think these monoclonal resistance mutations are actually induced by the use of monoclonals, or are they just sort of like, is that a proxy for immune escape more generally?
Yeah, I think it's probably the latter. But if we had more data, and I'm not really sure how widespread the use of the application of monoclonal antibody resistance is, it's certainly kind of a tantalizing idea. And those 2 cases that I mentioned where we did have breakthrough hospitalizations after the application of nirsevimab are interesting to test that. But again, it's a sample of 2, so we really weren't able to say anything too confidently about whether selection could have driven that. But if we see more, or I guess we're kind of ready for if we do see widespread breakthrough, to investigate that more.
Yeah, I mean, I guess, I mean, not to put you on the spot, but what would be your first step for figuring out a connection with severity? I mean, you have these data, and I mean, of course, when you're doing regression on a tree or like something like that, when you're trying to find an effect, there you have to be careful of phylogenetic effects and things.
Yeah, absolutely. And I haven't done work specifically like that in the past. So one, I'm open to ideas. I'm a sounding board for this. The second would be, if we did just see kind of a smoking gun, as we continue to, surveil the landscape. If next year we see, like if I go back to, the severity slides from the previous year. I think they're right before these. Yeah, here. So, again, I don't think it takes, even the most optimistic, regression, to say that, we see anything really clear. Again, the data is imperfect. This is not, this is a proxy for severity, but still, the severe cases are really distributed across the tree. At best, you could maybe squint on the RSVA tree and say maybe you see some clustering with, these lineages producing more severe disease, but we haven't run a model to test that. And I would be, I don't know, I'm not, I wouldn't try to make the case, based on this data, even if we had a model saying, these lineages are producing more severe infections. Now, if in the next year we see, a clade like this emerge and our cases are really tightly clustered with more severe outcomes, in this, in one particular clade, that would be, more of like I said, kind of a smoking gun situation. And maybe we could test that statistically and investigate it further. But I'm open to ideas. If anybody has any other thoughts on that, that would be wonderful. We've got the capacity to investigate some of that. So,
Well, it reminds me, like, there was work by Sebastian Bonhoeffer and Gabriel Leventhal about estimating set point viral load
okay.
in HIV. This reminds me a little bit of that. There was sort of like quite a discussion about that when they presented the results, but I can send you that link.
Yeah, that'd be great. I'm taking notes.
Well, I think we're at time. I really appreciate your contributions to public health and sharing your exciting science.
Yeah, thank you. And be sure to tune in. I know that the next two speakers are going to give great talks too. I know both of them, so
Cool.
I'm excited to listen to those as well.
All right. Thanks again.
Yeah, thank you.